The work I'm going to present today is our recent investigation of the role of Lysophosphatidylinositol, lipid second messenger, and this new link we found between the ABC transporter and the G protein couple receptor. Lysophosphatidylinositol is a lipid that is composed by a glycerol backbone that has a fatty acid and inositol head group. It belongs to a family of lysophospholipids. Among these, there are well-known second messenger like lysophosphatidic acid, the sphingosine 1-phosphate, and the role of this lipid is well known, and all the receptor and signaling is very well characterized. The lysophosphatidylinositol is mainly produced by the action of phospholipase A1 and 2, so that we can get lysophosphatidylinositol with the fatty acid in position 1 and 2. My early work some years ago identified LPI as lipids able to stimulate proliferation, especially in cancer cells. And what we found was that LPI was able to release calcium and in turn activate proliferation and in cancer cells. But also what we found was that lysophosphatidylinositol is specifically released by cancer cells. But what was not identified in those years was the mechanism through which the lysophosphatidylinositol is released by cancer cells, and also the specific receptor that is activated by lysophosphatidylinositol. Recently, a Japanese group has identified GPR55, a G protein couple receptor, considered a putative cannabinoid receptor, as a specific receptor for LPI. Several recent work has confirmed that indeed GPR55 is a specific receptor for lysophosphatidylinositol. So, therefore, we investigated the role of GPR55 as a specific signaling receptor for LPI. So, GPR55 is a G protein couple receptor that belongs to class A of G protein couple receptor as a sequence homology, low sequence homology with cannabinoid receptor 1 and 2, and has a higher sequence homology with other G protein couple receptor like the 35 and 23. What we found was that the ovarian and prostate cancer cell overexpress the GPR55, and to make a long story short, we found that the LPI is produced by the cytosolic phospholipase A2, is released in the media through the ABC transporter ABCC1, and once released in the media, it activates the GPR55 receptor present in the same cell. So we hypothesized then the existence of this autocrine loop in cancer cells. So we are now investigating the role of inhibitors of this loop, like ABCC inhibitors, as potential drugs to counteract cancer progression and development. And also, we're studying GPR55 antagonists like cannabidiol in vivo settings to see if the blockade of the GPR55 is indeed able to block the cancer growth in vivo system. So this will allow us to provide sufficient preclinical evidence that the blockade of this autocrine loop can block cancer progression development, so that we can bring this molecule to the clinical testing. So to summarize what I've said in this my short presentation, what we found is that the PLA2 produces LPI that in turn releases the ABCC1 in the media and activates the GPR55, then activates specific downstream signaling that leads to cancer cell invasion, migration, proliferation, and tumor growth. So I would like to stress the fact that so far ABC transporter has been seen as involved only in drug resistance. So this mechanism envisage new mechanism, a new concept for ABCC transporter or ABC transporter in general as molecule directly involved in cancer progression and development, so that the targeting of this molecule in specific setting could be used to counteract cancer progression. I would like to thank the people that work in my lab, and especially the Pancreatic Cancer Research Fund, Prostate Cancer UK, and Bowel Cancer for their support. Thank you.